Thank you so much for the opportunity to come to you today and speak about the American College of Rheumatology's COVID vaccine guidance that was recently released in the last 1 to 2 months. It's certainly a hot topic that I think is on mine and probably many rheumatologists and certainly many of our patients' minds. These are my acknowledgments and disclosures. I have the privilege to lead the ACR's COVID vaccine guidance task force and also to be part of the ACR's vaccine guideline task force, which is something separate that I'll speak to in just a minute. Polling question number 1. A 62-year-old man who has psoriatic arthritis is on methotrexate, 15 milligrams a week, and adalimumab at usual doses. He's soon to get one of the 2 mRNA COVID vaccines. It probably doesn't matter very much which one. Around the time of vaccination, you tell him to do what? 1, hold methotrexate for 2 weeks after each dose, continue adalimumab. 2, hold methotrexate for 1 week after each vaccine dose, also continue adalimumab. 3, hold methotrexate and adalimumab, both for 2 weeks after each dose. Or 4, hold neither methotrexate nor adalimumab and just go ahead and vaccinate them. Go ahead and vote. Polling question number 2. A 48-year-old woman with rheumatoid arthritis has been reasonably controlled on rituximab every 6 months. It's been 2 months since her most recent dose. She's also on meloxicam and methotrexate 10 milligrams a week. Her current CDI is 14, so that would be consistent with moderate disease activity, and she has a couple tender and swollen joints, and her physician and patient global are in the 4 to 5 range on a 0 to 10 scale. She's been offered vaccination with one of the 2 mRNA vaccines. In terms of the timing, you recommend for her to, number 1, get vaccinated immediately. 2, get vaccinated immediately, but hold the methotrexate for a week. 3, delay vaccination by 1 to 2 months, so that would put her getting vaccinated about 3 to 4 months after rituximab. Or 4, delay vaccination by 3 to 4 months, so vaccinate 5 to 6 months after rituximab. Go ahead and vote. Great. So the topic that we're going to speak to is the COVID-19 vaccine clinical guidance. So this is new and hot off the presses, and probably many of you have read this and hopefully did at least as part of their pre-reads for this session. So the nomenclature is rather important. This is a clinical guidance. It's technically not a guideline. What's the difference? So when the ACR develops clinical guidelines, it is a very extensive and methodologically rigorous, but very time-intensive process that typically spans more than a year. So there's a very comprehensive systematic literature review and a team of lots of people that's in charge of that. There are scenarios created that outline certain patient populations and what we might do to or for or with them, and then looking at outcomes as a consequence of those interventions that are informed by evidence. So it's quite laborious in that it undergoes lots of rounds of iteration, and the development process that typically spans more than a year was not felt to be feasible just in the interest of time and being able to say something about COVID vaccination in the near term. So for that reason, this is technically not part of a clinical guideline. The ACR calls these clinical guidances. It also does this when the evidence base is rather scant. So for that reason, that is the product that I'm getting ready to speak to you about today, and the PDF with the URL link is shown as on the slide on ACR's website. It has undergone one slight revision, so depending on when you looked at this last, it might have been changed a bit for some wordsmithing reasons and to add 1 or 2 things that probably are not material, but that might be incrementally informative that people had some questions or requested some clarifications about following the ACR's town hall. The composition of this clinical guidance task force is shown as on the slide. This is perhaps the most diverse in terms of specialty and subject matter expertise that the ACR's had on any of its task forces related to clinical guidelines or guidances. So there were 9 rheumatologists, those are shown in black font. There were 2 infectious disease experts, and then there were 2 either present or former CDC public health physicians with that expertise. This was really done in an attempt to recognize that, expertise in virology and public policy in relation to vaccination were an essential component of this whole process, and we wanted that diversity of perspective. What you also might notice, there's no patients that are on this. The ACR has increasingly recognized patients comprise a very valuable stakeholder whenever guidelines are developed. That will be part of the clinical guidelines that transcends all the vaccines, including COVID eventually, but was not felt to be feasible to include the patient members as part of this task force panel. The other thing I would point out is that there is a relative dearth of pediatric rheumatologists represented, and the implications of that will become apparent in just a few minutes as how the task force rated on certain things. The charge and the guiding principles are as shown. It was to develop clinical guidance for patient management on how to best use vaccination and to facilitate implementation of vaccination, not just in tandem with our offices and practices, but to think at a public policy level to the extent that that might be helpful in certain locales. It was based on scant but rapidly evolving evidence, and with the recognition there is almost no direct evidence for COVID vaccination and what happens in rheumatic disease patient populations. That evidence, of course, is expected to grow and accrue, but it was minimalist at best to begin with. For that reason, all of these guidance statements are considered conditional, which is ACR lingo to say, it's a good foundation, it's a good starting point for you and your patient to make decisions, but none of these are strong recommendations. Strong recommendation would typically imply that this is probably the right thing to do for most people most of the time. These are all conditional, meaning it's a great place to begin that conversation, but ultimately the font in red prevails. None of this is meant to replace your clinical judgment and to supersede shared decision-making between you and the patient. So this is a nice place to start that conversation, but not to end it, and you shouldn't look at it as overly prescriptive. The goal is to align with, but not to duplicate, other ACR efforts related to vaccine advocacy and some other domains that were beyond the remit of this task force. In terms of the literature synthesis, we generated an evidence report that was current through January 2021. We included all of the vaccine evidence that was synthesized for some of the prior recommendations that included vaccine statements. That would include the ACR 2012 and 2015 rheumatoid arthritis guidelines, the psoriatic arthritis guidelines that came out a few years later. So all the literature that was assembled as part of the systematic lit review for those efforts was drawn upon. And then for the mRNA vaccines, as well as the newer vaccines, to scour the gray literature or any other data that might be available. So initially we relied heavily on some of what was available on the FDA VRBPAC advisory committee sessions, where information was submitted by sponsors about their trial programs, et cetera. So this is not the peer-reviewed literature, because at the time those things hadn't yet been published in peer-reviewed format. And this was an iterative process that was continued through January 2021. So much of what I'm going to talk to you about didn't yet cover the new J&J vaccine, but revisions are both continual and ongoing. And this is expected to be a living document with a process that will continue over time and be updated. The first thing that we did following the assembly of the task force is to develop scoping questions. Essentially, what are the domains we're going to cover? What's in scope? What's out of scope? How much might we speak to things that the Center for Disease Control has already spoken to, or how much are we going to just reaffirm some of the things that they said? So those were the scoping questions. We divided ourselves into 3 teams. The first was focused on canvassing the data from clinical trials for the 2 COVID vaccines, as well as some that at least at the time were in development. The second work group covered both COVID epidemiology as well as vaccine effectiveness and how it relates to our immunomodulatory therapies for other vaccines. Influenza pneumococcal in particular, just because you're not going to find that in relation to COVID vaccination. And then the third work group focused on safety concerns related to, again, influenza, pneumococcal, shingles, et cetera, particularly around topics like disease flare, worsening, et cetera. This was all collated into a single-space 70-page evidence summary and iterated on a few times as new evidence accrued over the course of the work and was distributed to team members. The process used for this clinical guidance was one that the ACR has used in the past. It's the RAND UCLA modified Delphi approach. This had a couple rounds attached. There was asynchronous electronic voting on the statements that had been prepared that were, for the most part, positive affirmations of recommendations or background material that might be foundational in thinking about how to best use vaccination in our population. There was also a live webinar to discuss the ratings, resolve uncertainty. Where we might have been close to consensus, there was some real-time wordsmithing that was used just to reflect the diversity of opinions and perspectives to be able to achieve consensus. And for the most part, for all but 2 of the rating statements, consensus was achieved for a total of 74 rating statements. This is the high-level synthesis. This is again posted and has been updated at least once by the time you see this on the ACR website. You can read, and I'm not going to read all of this to you verbatim, but I pulled out some of the key things that might be helpful to highlight for you, and I put those in bold. So the second statement that honestly I was a little bit surprised was as controversial as it ended up being. So acknowledging heterogeneity due to disease and treatment-related factors, and after considering age and sex, patients with autoimmune and inflammatory rheumatic disease, that's this acronym AIIRD, and I'm going to heretofore just keep referring to this as autoimmune disease patients just because that's a mouthful and using that acronym is rather clumsy. So autoimmune disease patients are at higher risk for hospitalized COVID and worse outcomes compared to the general population. So those are actually 2 separate things. So you're at higher risk to develop hospitalized or serious COVID as an outcome, and then conditional on developing COVID that people seem to have worse outcomes. That can include, the need for intensive care unit stays or mechanical ventilation. That could include death or persistent infection. So in the publication that is forthcoming and might be out by the time that you see this, those were separate statements, but we've collapsed them mainly for simplicity as part of this summary. It was more controversial than I thought because many people said, well, I'm not even sure that this is true, that patients with our diseases have a higher risk for infection. Maybe they have worse outcomes, maybe they don't. Some of it might be related to comorbidities. Let me give you an example. Somebody with rheumatoid arthritis who smokes has interstitial lung disease. And because she has interstitial lung disease, if we accept that chronic pulmonary disease is a risk factor for bad COVID outcomes, you could say, well, it's not the RA that's her risk factor, it's the ILD that's her risk factor. And if she didn't have ILD and only had RA, maybe she's not at higher risk. However, in my example, the fact she has ILD is causally related to her RA. And so, in fact, you can't disentangle those things. So that was, in fact, the controversy, where there might be comorbidities that are either caused by, like in my example, or certainly influenced by some of our rheumatic diseases or their treatments. You can't very effectively disentangle the comorbidity from the disease itself. And so at a very high level, does it really matter why they're at higher risk? If in my example, the patient has ILD and that's what's putting her at risk, but that's related to her RA, or, in a different scenario, somebody with psoriatic arthritis and the metabolic syndrome, is on steroids for a time and, develops overt diabetes, et cetera, and diabetes is the risk factor for bad outcomes. Upstream of that was the fact that this patient had psoriatic arthritis and got a bunch of steroids, et cetera, that might be related to his disease. And again, you can't really disentangle, is the comorbidity truly separate and independent from the rheumatic disease and its treatment? And so that's where this voting statement ended up being crafted in the way that it did. So given that higher risk, and I'll show you a little bit of data to that point, autoimmune patients should be prioritized for vaccination before the non-prioritized general population of similar age and sex. So whether anyone listens to rheumatologists in terms of public policy about who should get COVID vaccination, you can answer that your own self. The idea behind this statement is reminiscent, or at least a bit of a proxy for what the CDC might term Category 1C patients, that due to a comorbidity that puts them at higher risk, that they should be prioritized. Now, I can tell you that not everybody is adhering to vaccine implementation according to those 1A, 1B, 1C, et cetera phases. And there was at least some thought that the CDC might not persist in that categorization and might come up with something different. So that's why this categorization of 1C isn't mentioned here, but it's the idea behind it that's relevant. Again, does this mean that autoimmune patients are at higher risk than 80-year-olds in nursing homes? No, of course not. And that's why there's this caveat about age and sex. A few other things in bold. So the expected response to COVID vaccination for many autoimmune patients on systemic therapies is likely to be blunted in magnitude and duration compared to the general population. So the vernacular is, the vaccine may just not work as well. That's the bottom line. I can tell you that there might be some unintended consequences, and some of my patients have been quite concerned about this and said, well, gosh, if the vaccine doesn't work as well and there might be some risk for flare, we'll get to that in a second, maybe I just shouldn't take it. That might be the logical interpretation, but in fact, that's incorrect. And the argument that I'll make is, let's say in clinical trials, the vaccine was, 94, 95% effective in the healthy volunteers. Let's just say hypothetically that it was only 70% effective for somebody with your condition. I made that number up. Nobody has that number about how well vaccination works in patients that are on immunosuppressive or modulatory therapies. But let's just say that it was 70% effective. 70% effective is better than 0% if that patient chose not to get vaccinated. So the point is that even though it might not be quite as good, if you're starting from a very high number, 70, 80, 90%, and it's somewhat diminished, that's still going to almost certainly be substantial enough for patients to motivate them to get vaccinated. So that's where talking about the magnitude of response, even if you're speculating, in my opinion, is helpful to facilitate conversations with patients to say, on balance, vaccination is almost certainly a good idea because some protection is better than no protection if you forego vaccination. The last statement I want to speak to, a theoretical risk exists for flare or disease worsening following vaccination. That has been evidenced in what are mostly case reports from other vaccines. So again, is it a risk? Sure, it's a potential risk at a minimum. However, the task force rated that the benefit of vaccination for rheumatic and musculoskeletal disease patients outweighs the potential risk for new onset autoimmunity. That could be new onset symptoms or manifestations related to their disease. That could be other things, Bell's palsy or Guillain-Barré. A few cases of that have, in fact, been reported. So that is a legitimate concern. In the Moderna trial, there was one case of new onset rheumatoid arthritis that investigator judged as probably being related to vaccination. That said, in trials of tens of thousands of people, there's single-digit case reports at this point. I'd also be quick to remind people, there have definitely also been case reports of new onset autoimmunity after COVID infection. Not vaccination, but after COVID infection. So I remind people of this, that simply getting infected, even though you might feel crummy or have sort of long hauler type symptoms, that people can develop new onset autoimmunity. There's been published case reports of bona fide vasculitis and rheumatoid arthritis following COVID infection. So lest one be concerned that this is only a vaccine safety issue, we see this even after COVID infection, and that needs to be taken into account as people are weighing the pros and cons of vaccination. Let me give you a little bit of population-based data that is countrywide and rather lacking in the United States, but in countries like Denmark, where there can be national registers that link people cradle to grave, you can do certain things a little bit more nimbly than maybe we can do here in the US with that same speediness. So I'm showing you a paper that at the time this is recorded is still in press, so apologize for the formatting, but I've circled the most important element. So the column headers, they looked at all inflammatory rheumatic diseases, and the bulk of the data is for people with rheumatoid arthritis. And I've circled in red the incidence rates of per 1,000 patient years of hospitalization with COVID infection. So maybe the most important column is the second one. It's the rheumatoid arthritis column. That's the 1.97 versus the general population in the rightmost column that I've circled, 1.26. So if you then say, well, let's control for age and sex, what you end up with is that rheumatoid arthritis has a 1.84-fold increased risk in hospitalized COVID infection compared to the general population. There's at least 5 studies that I know of that have substantial sample size that is beyond single center experiences that suggest that, somewhere maybe in the 1.3 to 1.8 fold range, we see these estimates for people with rheumatology conditions, RA probably being more common among them. But there does appear to be an increased risk based on population studies. In this particular one, even though they're able to look countrywide, there are still relatively small numbers of cases for people with RA or other rheumatic diseases. But nevertheless, it does seem like that this paper and some that are referenced in the forthcoming publication in arthritis and rheumatology do seem consistent with the fact that COVID is more likely to occur, at least hospitalized COVID, in people with certain rheumatic diseases like RA. So several other guidance statements, and maybe the most important one to message to patients. So rheumatic and musculoskeletal disease patients and autoimmune patients should get vaccinated. Hard stop. So yes, the vaccine is recommended. In fact, this might seem so obvious that it's milk and apple pie. On the other hand, at least for a time in Canada, the Canadian provincial authorities said, well, gosh, rheumatic disease patients on immune suppressing therapies weren't represented in the trials. Those people shouldn't be vaccinated, which you might say, gosh, that seems a little odd. And at a minimum, those people should be prioritized because at least some of them are at higher risk. But in fact, at least for a time, they were prohibited. So without saying things like this, bad things can happen like that. I can also tell you that there's a large pharmacy chain in the U.S. that has been saying that if you're on any biologic, you shouldn't get vaccinated without having stopped for more than 90 days. That might be relevant for a live virus vaccine, none of which these are, not even the newer ones, and certainly not the mRNA vaccine. So we've definitely seen examples in the United States of patients getting refused vaccination. So hopefully these sorts of guidance statements will help. The second bolded statement here that I've listed, patients that don't have an autoimmune disease, but if they're on immunomodulatory therapy, even off-label, should be vaccinated in the same fashion as anybody else that is represented in these guidelines. So for example, if you have a patient that you're using, say, methotrexate to reduce the immunogenicity of pegloticase, you would just follow the methotrexate specific guidance statements, even though arguably, gout is not quite of the same magnitude as is RA or lupus or vasculitis in terms of, some of the immunomodulatory or suppressive treatments they might get. But the fact is, if they're on the same medicines, then you should be thoughtful about that as well. The third statement, based on the data for the mRNA COVID vaccines in the U.S., at least at the time, there's no preference for one over the other. You should just get whatever's available. These are specific to the 2 mRNA vaccines. Of course, with the newest one that is available under the emergency use authorization, it is not represented here. The task force is currently revising their statements. If I were to speculate, I might guess that they will feel quite similarly that, whatever is available to you probably is the one that you should get, and don't delay waiting for one that might be perceived to be better, but probably it isn't, or at least is far from certain that one might be better than another for whatever reason. So get what's available. And then finally, and perhaps among the more controversial statements, and the last one in bold on this slide, midway down, so healthcare providers should not routinely order any lab testing to assess immunity post-vaccination, is my patient protected, or to assess the need for vaccination in somebody who hasn't yet received it. So why is that? So remember that you can test for antibodies to a couple things. You could test for spike, which is what you would want to following post-vaccine, but that's actually not what some of the commercial assays measure. They measure nucleocapsid, and that might make sense if you're trying to detect infection, but not so much vaccination. And so for many people, they got the wrong test if what you're trying to do is assess post-vaccine response. So that's one important issue is, are you even ordering the right one? The second is, even if you're ordering antibodies against spike following vaccination, I think we would argue that there's not a whole lot of evidence about what you should do with the number. There's not a lab correlative protection. You can't say, oh, if you're at this level of an antibody response with XYZ assay, that means the patient's protected. That would be a so-called lab-based correlative protection. Mrs. Smith, your number was whatever, so you're protected. We don't have that. We don't have science that suggests that. So, the task force probably would contend, I certainly would, that we don't yet know what to do with the number, but it certainly would probably be beyond the evidence to tell Mrs. Smith that she's, protected and thus reassure her, because we don't have that relevant for individual patients. At best, we might soon have that for groups of patients, but nevertheless, at this time, we just don't have the science to support that in terms of routine clinical management. On the other hand, I definitely can see that that will accrue, and there certainly might be settings that this would make a lot of sense. We just don't have that yet. In terms of holding medicines, for the vast majority of our therapies on the first two large rows here, there was no recommendation to either hold the drug or modify vaccine timing. The only exceptions were for methotrexate, Janus kinase inhibitors, and sub-Q abatacept. There was a recommendation to hold methotrexate for a week after each dose for those with well-controlled disease. That was based on the Korean RA trial that did this, that found a better immunogenic response to influenza vaccination if you held methotrexate. Now, when they did that, they held for 2 weeks, not just 1 week. So you might say, well, why was then there a recommendation to only hold for a single week and not 2, like in that trial? The reason in part was related to the practicalities. If you're trying to give a 2-dose vaccine, you hold methotrexate for two doses, and then you might give 1 methotrexate dose in the middle, and then you get your second mRNA vaccine and hold for another 2. Now the patient has been off therapy for 4 weeks, and that's just a complicated, confusing set of instructions. So mostly for practicality and simplicity, this was the recommendation, just hold for a single dose. With the idea that Janus kinase inhibitors, and because of their profound effects on interferon signaling, might attenuate vaccine response, there was a recommendation to hold for just 1 week. They obviously have quite short half-lives, and so that was felt to be a biologically relevant window. And then finally, for abatacept sub-Q. So this is a little bit different. The recommendation was to hold for a week before and after just the first dose of the vaccine. Why is that? Well, the reason for that and just the first dose is that priming a naive T cell to recognize a virus that it's never seen before is critically important. That first date or that handshake needs to go well, and abatacept that can block other vaccine responses, particularly for an antigen that the immune system has never seen before, unlike flu or pneumonia, where every vaccine we give is mostly just a booster of an infection we've seen something like in the past. Not so with SARS-CoV-2. It was really the priming of the naive T cell that was felt to be most important in relation to the abatacept sub-Q timing considerations. A few other statements and timing around for abatacept IV and cyclophosphamide. People can read that. The rituximab bears a little bit of mention. So the recommendation, based upon mostly studies with humoral immunity outcomes, was to schedule vaccination so the vaccine series was initiated about 4 weeks before the next cycle. So if you're giving it every 6 months, you would start the vaccine series roughly about 5 months into it. So that's the guidance, although there's some caveats about that. The caveats around several of the things I just described are shown mostly on this slide for context. A few of the foundational principles are shown here, a couple in bold. The one in the middle, there's no direct evidence about vaccine safety or efficacy. All the timing considerations I've just described are predicated not on safety concerns, but on trying to optimize vaccine response. Also, there's no reason to expect vaccine harms will trump benefits in rheumatic patients. And then the second bolded statements, the risk of deferring vaccination and failing to mitigate COVID risk should be weighed against a possible blunted response to the vaccine if you give it under suboptimal circumstances. So for example, patients on 40 milligrams of prednisone has gotten rituximab, 2 months ago and has poorly controlled disease. Should you vaccinate then, or should you wait a couple months hoping she's on less steroids and is further out from her rituximab? There's pros and cons both ways. There's not a right answer. Some protection is better than nothing. If she can do physical distancing and is at low risk otherwise, then maybe she can wait. On the other hand, if she's a healthcare worker, a teacher, and is constantly exposed, maybe she can't. So this, again, is where there's a judgment call here that you need to balance the, should I delay or should I do it now? In general, the task force recommended that now is probably better for the vast majority of people. There's a few myths or assumptions that I've collated from a variety of mostly patient sources that I would urge you just to think about, and we can talk about this in some of the Q&A. Hopefully some of these positive guidance statements give you some armamentarium about how to respond to some of these. Myth number 3, I have an immune condition, the vaccine won't protect me. Again, we don't have direct evidence yet, but there's no concern that this should make you sick like a live virus vaccine would have concerns for, even with the new adenoviral vector vaccines. And then a few other things about flare that, again, we don't have a lot of data. The vaccine trial for the influenza vaccine, 11% of people who held methotrexate 2 weeks flared using the DAS and worsening of the DAS by more than 1.2 units versus 6%. Is 11% higher than 6%? Sure, but it's not that common overall. And that's for flu vaccine, but not a huge difference regardless. In terms of next steps, the manuscript with much more detail and all the citations is expected to be published this month and might be out already in arthritis and rheumatology. The task force is in the process of updating this for some of the newer vaccines, as well as new evidence for the existing vaccines. And of course, we need direct evidence for this. Eventually, all of this work of this clinical guidance task force will be part of the larger ACR vaccine guideline task force that I and many of the others are part of. And then finally, several of us are working on one of the several vaccine studies that would accumulate direct evidence in rheumatology patients, where we wouldn't randomize people to placebo vaccine, but rather we would come alongside them and get blood and assessments, in patient diaries and electronic case report forms on a smartphone app to collect information about safety, reactogenicity, disease flare, things like that. And then there's a nested ancillary study where we randomize people to temporary hold therapy for some of the short-acting agents that there's current statements that suggest that might be a good idea, but we just don't have that evidence. So thank you so much for your interest and your attention, and I'm glad to field any questions in the Q&A.